Welcome back. So this time we're going to talk about the idea of transcription and translation, or also we refer to it as the central dogma of biology. My handwriting's so much slower, it makes it less dramatic, but it's actually a pretty big deal. All right, let's get started. Right, so the basic idea is we're going to take information from DNA, we're going to write it down on some RNA, just like part of a gene that we're interested in, right? And then we're going to take that RNA and use it to make the protein that we need, right? The amino acid sequence. That's basically it. But there's a couple of steps involved. So the idea is all
of the information it takes to make you is stored on your genome, right? So this is your DNA genome. But say you only need to know about, for this case, you've got a paper cut. And so you just need to take this little piece of DNA from this area right here, which is going to tell us how to do things like repair your stem cells, and also maybe some capillaries, and some stuff like that. And we're going to replace that part in your finger. So you don't need the DNA on all the time, you just need that information. What's going to happen is we're going to make little RNA transcripts. So the RNA is going to be making copies of that area, and I'm going to make a lot of these little copies, right? So this RNA is also going to be moved down to the area where we plug it in and have a couple of different types of RNAs. These are transfers, so this will be messenger RNA. mRNA. This is going to be transfer RNA, right? Because what it's going to do is it's going to transfer different types of amino acids so that we can join the sequence together eventually when we actually need it with a ribosome. So ribosomes look like little hamburgers, and they're actually little machines. And what they do is they take the mRNA and they read it. And then as they're reading it, they're going to grab some of these tRNAs, and they're going to start making a little chain, right? So they'll make a chain of all the sequence of amino acids we need to have that particular, say, stem cell or capillary or whatnot replaced in your body, okay? So that's the idea. Let's take a closer look with some much better graphics.
Okay, here we go. Central dogma. So we're going to take DNA, we're going to make transcripts. First they're going to go to mRNA. We're going to use a couple of other types of RNA and translate that information on the DNA to a final three-dimensional protein that we can actually use to maybe repair some cells or to make some new structures. That's how it works. So here's a little bit closer look at just the mRNA part of the process. So here's our DNA right here. And here's our mRNA right here. And actually, this is pre-mRNA. mRNA is going to go through a couple of different maturation stages. So as you can see here, we have our DNA, and we split it open, right? We split it. Okay, this is a replication fork. And then we're going to allow an RNA polymerase, which is an enzyme that makes polymers, right? Polymerase. See what it did? See they named it? And now you know about polymers and then ases, and there it is. Right, polymerase. And so what that polymerase is going to do is it's going to read all of the specific sequences, and it's actually going to put a complementary base pair down and make a single strand, right? So this is a single strand of mRNA. How nice. Isn't that nice? And so after it's been prepackaged and exported out of the nucleus, right? So all of this happens in the nucleus. And then this part is shipped out of the nucleus to the cell. And waiting, usually, and it's parts of the cell known as the endoplasmic reticulum, or just around the outside of the nucleus, are the ribosomes. Right? So here's a ribosome. Ooh, that's terrible. Let's just fix that. Ribosome. There it is. Okay, so ribosome highlighted. Okay, so here's your ribosome. The ribosome, like you said, is going to attach to it. The ribosome also has a little bit of RNA too, and it's going to read that sequence, and as it reads it, it's going to create this polypeptide chain, right? I remember when we talked about protein folding in video 4, so things like primary, secondary, tertiary, and quaternary structure, we move forward from there. Here's a close-up look at what happens in the cell, right? So I'm showing you a lot of graphics because sometimes it's hard to get your head around these ideas the first time, so maybe if I just explain it a few different ways, you'll be able to visualize it. So that's why we're kind of going through it a few different times. Alright, so here's that nucleus, right? So part of it happens in the nucleus and then some of it is transported out into the cell. It floats around in the cytoplasm, right? So this is where structures like the endoplasmic reticulum, which we'll talk about in future videos, or just freely floating ribosomes will be waiting. So what happens is we have, remember, RNA polymerase. It's going to make a polymer of RNA, right? It's an A's. So it reads the DNA sequence. Double-stranded DNA molecule is opened or unzipped. So we're able to read it. We make a single-stranded mRNA molecule from it. We call it an RNA transcript. We process it a little bit, which we'll talk about later. For us in eukaryotes, we actually do have introns and exons, so we need to splice out some of the parts of the RNA that we're not going to use and join the other ends back together. We also need to cap and put a poly A tail on these transcripts. That helps protect it during transport out of the nucleus and into the cytoplasm. So once it's transported into the cytoplasm and out of the nucleus, we're then able to be taken up by an RNA molecule, I'm sorry, not a molecule, but a ribosome that has our RNA, right? And so what it's going to do is it's going to find these transfer RNAs. So here's a transfer RNA, here's a transfer RNA. We refer to them as tRNAs, okay? These transfer RNAs have little tiny amino acids onto them, right? So here's an aminoacetyl tRNA synthetase. And so that's that's an enzyme that attaches the amino acid to the particular and specific transfer RNA. Something important to look at. See, each one of these has a different sequence. ACC, AAA, UAC. That means that you can put a different amino acid on each one of these different transfer RNAs. The other thing is, it'll tell you which one it's going to be, right? These are anticodons. These down here are codons, okay? So these are 3-letter words that tell us which particular amino acid needs to go in what placement in the sequence. It's also how we can ensure that that particular amino acid goes into the right place every time. Clever stuff.
So there are 3 types of RNA. First one we talked about is called messenger RNA. The second one is ribosomal RNA, and the last one is transfer RNA. Messenger RNA is going to be carrying coded instructions for protein synthesis, carries them right from the DNA molecule. Transfer RNA, or tRNA, in this case they have RNA-T, which is fine, carries specific amino acids to the ribosomes during protein assembly. And remember that anticodon-codon pairing is going to let us make sure that we actually put the right one in the right sequence. All right, check. And finally, part of the ribosome is actually made of rRNA, or ribosomal RNA. It speaks to how old this ribosome actually is. It's probably one of the first functional proteins to ever evolve in living organisms on this planet.
So remember we were talking about codons and anticodons? So those are very, very important. Those are the little words that tell us exactly where we need to place each one of the amino acids in the sequence. Remember, if an amino acid sequence is out of position, then the protein will not fold correctly, and someone can end up with a very serious disease state, or even a minor one. Whatever it is, it's still not usually going to be super functional. So, I just want to take a look and see this. This is AUG. This is what we call the MET, or start codon. It tells us this is the beginning of the sequence. That means every 3-letter grouping after that is going to be responsible for the placement of another very specific of the 20 amino acids. Now, why is it so important that they're all in the correct position? And how is it that even one little amino acid can really mess anything up? Well, here's how. Here's some of the structures of the amino acids. This will kind of let you know what you're dealing with. So we have some amino acids that are on the acidic side, others that are considered basic. some that are neutral and nonpolar, but others that, remember, are polar. Do you remember that from when we were talking about water? All right, so it means that they can sometimes either bond together or they can repel each other apart, and they can, just a single amino acid can change the overall structure of that protein, meaning it can denature it or it can make it too strong. And so that's an important thing. Each one of the proteins that are put together in all of your cells are predictable. They work together just like a puzzle piece. Have you ever had a Lego that just didn't fit right? How frustrating that is? Well, if that's one of these essential amino acids in a protein, then that can really disrupt the overall structure of whatever type of cellular component you're trying to make. So even a single amino, or even a single actually base pair change resulting in an amino acid change can create a disease that's very serious. So one of the ones that's actually used in your textbooks that you can read about, so it's in Pearson's and the OpenStax and any of these textbooks, in the McGraw-Hill as well, we're going to talk about sickle cell anemia, right? So that's an important different disease state that's just resulting from the change of one single amino acid. All right, now hopefully that gives you a little bit of perspective on the situation, but feel free to look up some other ones on your own. If you have any questions, also email me. I love helping.
This is the codon chart. This will help you figure out which specific amino acid you're coding for. So for these, you're going to be looking for the first, second, and third letter. Remember, there's always three. There's always three, right? So if we were going to find UAA, what we would do is we'd start with our first letter over here. We'd find U. We'd go over to the A's, so this is the A group for that U, right? Okay, and then we'd look over here at the third letters, and so the last one is A. So, oh, what do you know? It's a stop codon. Okay, so this is how we use the chart. So if you'll notice, some of these show up a lot. Others don't show up quite as often, right? But it helps us become very specific. It's a little bit redundant, so you might hear about the redundancy of the codon chart. You might also see it in a wheel shape. Personally, I like the chart, but some people really swear by the wheel. But whatever works for you and is the most accurate for your reading is the one you should go with. So we spent some time talking about transcription and translation. So transcription is the process by which we're going to make a single-stranded RNA molecule that's just over one particular gene or one particular locus of a region in the genome, right? So we don't have to make a transcript of the entire DNA every time, just the parts we need. We can make a lot of copies, right? Tons and tons and tons and tons of copies. After we process them, we can ship them out of the nucleus. Once they're out of the nucleus, they meet up with these ribosomes. These ribosomes read them and put the correct tRNAs in order to make the exact growing polypeptide chain or amino acid sequence that we need to eventually fold into primary, secondary, tertiary, or perhaps even quaternary structure and then continue on with whatever kind of repair, growth, or whatever kind of mechanism that the cell needs. All right, I hope that was helpful.